Good morning, ladies and gentlemen. The topic of today's lecture is very interesting and, of course, very important for understanding the genetic transmission of various diseases and the genodermatosis. So the topic of today's lecture is the principles of genetic transmission of diseases and genodermatosis. First of all, some words about online Mendelian inheritance in man, that is abbreviated as OMIM. This you will see while you are reading various genetic disorders. It's a catalog of 6,000 known human Mendelian characteristics with skin manifestations and was created by Dr. Victor of Johns Hopkins Hospital, Baltimore, USA. OMIM is an excellent starting point for acquiring up-to-date information on human Mendelian characters or phenotypes. Each character is given a 6-digit MIM number, which is widely used to identify inherited disorders in medical literature. So this is important in catalogs and in cataloging the diseases and in keeping a record, because these 6-digit numbers will be uniform throughout the world. What is a familial inheritance? It refers to the clustering of a disorder with more close relatives affected than predicted by population prevalence of the condition. That is, more close relatives are involved with the disease than expected from the population prevalence studies. For example, 25% of patients with psoriasis have an affected first-degree relative, but the pattern of inheritance is uncertain. So, psoriasis can be called as a familial disease because there are more chances of family members being affected with psoriasis with no particular type of inheritance. And this is also common in other dermatological diseases like atopic dermatitis and in androgenetic alopecia. Inherited disorders. The disorders that require the transmission of genetic variants from one generation to the next. So there is a genetic defect, and then the same defect is transferred to the next generation, then such disorder will be called as the inherited disorder. What's a congenital disorder? It simply means that the character was present or detectable before birth in utero. Such abnormalities may not be genetically determined and include the developmental defects due to environmental infections, for example, rubella, deformities arising from physical insult, for example, amniotic band, or many drugs which causes teratogenic effects, for example, isotretinoin. So such disorders will be detected before birth and are called as the congenital disorders, and they may or may not be genetically determined. Genodermatosis. Of the inherited skin abnormalities, the largest group is the single gene disorder, which require an alteration in function of a gene and are known commonly as genodermatosis. So the genodermatosis, you can identify that particular single gene defect that is the cause of all the abnormalities which we see in such diseases. What is a multifactorial inheritance? Predisposition to more common skin disorders, including atopic eczema and psoriasis, is determined by action of more than one gene, with significant evidence of further modulation by environmental factors. Such disorders display complex pattern of clustering in the families and are referred to as multifactorial. So the multifactorial disorders are those disorders in which different genes and different environmental influences affect the causation of that particular disease. Such examples, the example include atopic dermatitis and psoriasis. Chromosomal abnormalities. The chromosomal abnormalities either in number, which are called as aneuploidy, or form, that is translocation, complex rearrangement, or microdeletions, are important to the clinicians, particularly if patient presents with range of abnormalities, including mental retardation. So the chromosomal abnormality disorders are common and have common skin manifestations as well. So such chromosomal disorders can be because of some disorder in their number, either less number of chromosomes or more number of chromosomes, or because of translocations and microdeletions, which results in certain peculiar phenotypic abnormalities that include particularly the mental retardation. Then gene. What is gene? A gene is a sequence of bases in DNA encoding a polypeptide. The precise position of the gene on a genetic map is called as its locus. Two copies of every gene exist in a human. One is a maternal copy and other is a paternal copy. In all 22 pair of chromosomes, and in both the X chromosomes in a female. This does not apply to male because the sex chromosome in male are X and Y, which are dissimilar chromosomes. Alternate genes on pair of a chromosome at a single locus are called as allele. So it is said that for every disorder, there are two genes, one from the father, one from the mother, and the single locus where these genes are located on pair of chromosomes is called as the alleles. An individual with two different alleles in a particular locus are heterozygous. If the two genes that are inherited from both parents are different, then that individual will be called as the heterozygous. When both alleles are identical, the individual is called as the homozygous. So both the genes inherited from the parents are similar, then that particular individual is homozygous for that alleles. Gene locus on X chromosomes are not one of a pair in males, and when such genes is abnormal in male, the term is hemizygous is used. So a particular gene, if present on X chromosome of a male, and of course the similar copy is not seen on a Y chromosome, then that particular individual or male will be called as the hemizygous for that particular allele. Dominant and recessive. An allele is regarded as dominant if it manifests as a phenotype when present on only one member of chromosome pair, that is heterozygous state. The other one is not the similar gene, and will be called as a recessive if it must be present on both chromosome loci, homozygous state, before it can exert its full effect. So the first important point is, for every dominant gene, there has to be a recessive gene on the other locus. So the two alleles are not similar. They are heterozygous. And one of the genes is received from one of the parents. So such heterozygous alleles, when one of the allele always manifests as a phenotype, that it gives a phenotypic abnormality, an individual will be called as the dominant allele. And for recessive alleles, it is must that that there should be a corresponding recessive allele on the other received from the other parent. So for recessive characters, the individuals are usually homozygous, that is receiving the defective or abnormal gene from both parents. Only then can it exert its full effect. So here comes the family marriages which results in inheritance of abnormal recessive genes in a person, in an individual. Hence, it is apparent that the term dominance and recessive refers to a phenotypic characteristics rather than a gene. So it's obvious that if a gene is manifest as a phenotypic character, only then it is called as a dominance or recessive trait. Autosomal or sex-linked. Those genes that are born on chromosomes other than sex chromosome, that is X or Y, are known as the autosomal genes. And the characters that are controlled by genes born on X and Y chromosomes are termed as the sex-linked characters. The Y chromosome is much smaller than X chromosome. So the great majority of sex-linked genes are exclusive to X chromosome and having no active counterpart on Y chromosome. For these traits, the term recessive applies to male who only carry one mutant allele so that if a male has inherited a particular sex character on X chromosome, which does not have a corresponding allele on the other X chromosome, because which is of course not present in a male, then even if that character is a recessive character, it will always manifest. In females, since the sex characters will be corresponding on the two X chromosomes, so the females, such kind of characters will not be obvious. So the females who carry an X-linked mutation are called as the heterozygous. Y-linked genes. Genes that are Y-linked are primarily involved in male sex determination. So there are only a few sex characters that are determined on Y chromosomes, but that are particular for males only. For example, SRY gene is controlling the testes determining factor or spermatogenesis. Hairy pinna is another phenotype that is Y-linked. Mitochondrial gene. In addition to the nuclear DNA, mitochondria contain a circular chromosome comprising of 14 protein coding regions. The mitochondrial DNA codes for enzymes involved in respiratory chain and oxidative phosphorylation. So the mitochondrial disorders are essentially muscular, neurological, and ophthalmological diseases, and transmission is almost exclusively matrilineal, that is from the mother lineage. So the mitochondrial disorders are a few of the disorders that are mainly transferred from the mother side, and these are results in various neurological, muscular, and ophthalmological manifestations, and are mainly coding for particular enzymes that are deficient in these molecular gene disorders. Pedigree patterns. An understanding of a characteristic pedigree pattern of rare simple inherited Mendelizing disorder is essential for counseling the prospective parents on risk of having an affected children. The three main inheritance pedigree patterns are autosomal dominant, autosomal recessive, and X-linked recessive. There is a fourth one which is called as X-linked dominant, but that's quite rare. So, by knowledge of whether this disease is transmitted as autosomal dominant, autosomal recessive, or X-linked recessive pattern, forms the basis of genetic counseling in the affected parents. First of all, the autosomal dominant inheritance. The characteristics of autosomal dominant inheritance. Autosomal dominant disorders affect both male and female. So since the disorder is on the autosomal chromosomes, on somatic chromosomes, so both males and females are affected. The affected individuals are heterozygous for the abnormal allele. As I've told before, that an autosomal dominant character will always be in a heterozygous manner, and the other allele will be a non-dominant character. So the person need not have a homozygous character for the autosomal dominant pattern to manifest. Only one of the gene in the allele is sufficient to manifest as autosomal dominant disorder. Affected person will usually have an affected parent, apart from recipients of new de novo mutations. So since it's a heterozygous character, 50% of the offsprings of affected parent will be carrying that particular gene and will be affected. The age of onset and severity may be variable, and disease may not manifest until the adult life. Some autosomal dominant disorders show reduced penetrance. That is, a person who inherits the gene does not develop the disorder. So there is another term introduced here, that is reduced penetrance. So it is not must that autosomal dominant disorder, if present, will always manifest as a phenotypic abnormality. So there can be less manifestation if there is reduced penetrance. So examples of autosomal dominant disorders include acute intermittent porphyria, epidermolysis bullosa, some forms, ichthysis, some forms, neurofibromatosis, tuberous sclerosis, hereditary hemorrhagic telangiectasias. Autosomal recessive inheritance. Autosomal recessive inheritance is again autosomal and is seen both in male and in female. So there is no particular sex preference for such disorders, as well as autosomal dominant disorders. There is no sex preference. As I've told before, the autosomal recessive disorders need to be homozygous. That is, both alleles should be carrying the same gene from the two parents. And here, the familial inheritance is, family inheritance or family marriages are important. On average, 25% of the children of heterozygous parents will be affected. So the parents definitely are not manifesting the disorder. They will be carrying heterozygous and will be carrying one of the gene, abnormal gene, and one of the dominant gene, which is suppressing a particular phenotypic abnormality. So this is seen in both the parents, in mother and father. And what happens that in 25% of the offsprings, that is, if they give birth to 4 offsprings, that is 25%, if they give birth to 4 offsprings, there is a chance that in every offspring, there is a 25% chance of developing an autosomal recessive disorder. And it's not that among the 4, 1 will be affected and 3 will be not affected. It is possible that all the 4 may be affected, and it is possible that all the 4 may be healthy. So 25% of the offspring have a chance of inheriting both the recessive abnormal genes from the parents, and when these genes become homozygous, it will manifest. Typically, no family history is seen. The parents are usually normal. Consanguinity increases the risk of autosomal recessive disorders. The offsprings of an affected person will be healthy heterozygous and can be affected only if the other parent is also a gene carrier. So if there is a particular disease in a family and an individual is absolutely normal, or even if it's suffering from the disease, that is an affected person, if say the affected person is a homozygous for that particular gene, for that particular disease, if that individual marries a normal person and that normal person carries the dominant genes which are disease-free, then in the offsprings, that dominant gene will stop the manifestations of the recessive gene, and the disorder will not be manifest. Autosomal recessive disorders are often severe. For example, inborn error of metabolism. Examples of autosomal recessive inheritance include epidermolysis bullosa, severe form, ichthyosis, more severe form, acrodermatitis enteropathica, phenylketonuria, xeroderma pigmentosa. X-linked recessive inheritance. Usually only males are affected because males will not be a heterozygous dominant suppressing gene for that particular character. The disorder is transmitted through healthy female carriers. Occasionally, a heterozygous female may show some features of the condition as a result of non-random X inactivation that I will explain you later. So usually the females will not manifest, but some forms may be manifest if one of the X chromosome is inactive. A female carrier will transmit the disorder to half of her sons, and half of her disorders will be carriers. Since the daughters will be carrying another X chromosome that will be carrying a dominant gene that would be suppressing that phenotypic abnormality. So the the daughters will be the carriers. But in males, if they inherit that disorder, there will not be a suppressing allele, so they will manifest the disease. The trait cannot be transferred from father to son because it's an X-linked trait. So the inheritance will not be seen from fathers to the son. However, it can be transferred to the daughters. An X-linked recessive condition should be considered when the family history indicates affected males in different generations of same family. Family history is not always positive, as new de novo mutations are common. Examples include anhydrotic ectodermal dysplasia, Fabry's disease, Menkes syndrome, and ocular albinism. X-linked dominant inheritance. X-linked dominant disorders will give rise to disorders in both hemizygous male and heterozygous female. So since it's a dominant character, only if it will be heterozygous even in female and will be manifest. And of course, in males, there will be no other corresponding gene. Affected males will transmit the disorder to their daughters, but not to their sons. Affected females will transmit the disorders to half of the sons and half of the daughters. In some disorders, the condition is lethal in hemizygous male, so only female patients are encountered in clinical practice. So such disorders are of severe intensity, and in males, there is no corresponding allele which reduce the intensity of the disorder. So such disorders are usually lethal in male. While in female, even with presence of such disorders, the corresponding allele reduce the effectiveness or severity of the disorder, and that's why these females survive. Examples include incontinentia pigmenti and X-linked form of orofacial digital syndrome. Then penetrance. We used this word before. Penetrance is the degree variability in expression of a character in question and the frequency with which a gene produces any effect at all is measure of its penetrance. So a gene would be non-penetrant if it does not express phenotypically. So presence of a gene in an individual is not must that such individual will manifest its phenotypic characters, because the phenomena of penetrance shows that such character may not be able to manifest because of reduced penetrance. Expressivity. This defines the degree of expression or severity of a trait. It is a designation of qualitative and quantitative manifestations of a specific genotype and is the variability in the phenotype produced by a given genotype. So now the question arises in expression of a particular gene, and expressivity is a measure of qualitative and quantitative phenotypic manifestation of a particular gene. Patients with same mutations may have differing severity of the disease. This may be due to differing environmental influences. For example, differing amount of exposure to X-ray radiations or UV radiations. So two individuals may be carrying a certain gene defect, but both will not be expressing the same amount of phenotypic abnormalities because the two may not be having the same environmental influence as the other individual. For example, one person is exposed to X-ray radiations and other person is not. One person is an outdoor worker and other person is not. So the person who is more exposed to the environmental influence will express the gene more as compared to the other individual. Pleiotropy. The multiple phenotypic effects due to the primary actions of an abnormal genotype is referred to as pleiotropy. So single autosomal gene, such as those from a Gardner syndrome, from Pachyonychia congenita, or neurofibromatosis, causes multiple phenotypic effect in several organs. If we take example of neurofibromatosis, it results in neurofibromas, plexiform neurofibroma, Caffolet macules, freckling, and mental retardations. So there are multiple phenotypic abnormalities that are associated from a single gene defect. And such multiplicity of the phenotypic characters arising from a single gene defect is called as the pleiotropy. genetic heterogeneity. Genetic heterogeneity is the term used to describe clinically similar disorders, phenotypes that result from different genetic defects. The concept of genetic heterogeneity is important for several reasons. Disorders that appear the same can be due to entirely different molecular defect with different natural history and requiring different treatments. Defects may have different inheritance patterns, plays significant role in common complex disease states. So genetic heterogeneity means that one disorder that manifests in an individual, different individuals have different bases of inheritance. For example, a disorder can be transferred to an individual's as autosomal dominant or as familial inheritance or due to environmental influence, or a single disorder can be transferred as autosomal dominant, autosomal recessive, etc. So there are multiple disorders which you will see in the text which show the phenomena of genetic heterogeneity. The significance of this phenomena is that there will be different treatment options for a specific causation of that particular disorder. If it is an environmental influence or if it's a genetic influence, then there will be different treatment options, different counseling skills, different precautions or preventive strategies that you will formulate with that patient for that particular disorder. So before going for genetic counseling and counseling for a particular disease, it is must that one may be able to determine what is the specific pattern of inheritance of that particular disorder. Mutation. The term mutation has traditionally been applied to the genetic variations causative of a Mendelian disorder. Most mutations are spontaneous and unexplained. However, certain factors such as mutagenic chemicals and ionizing radiations can increase the rate of mutation. If a mutation occurs in a somatic cells, it's called a somatic mutation. Only the descendants of that cells are affected and there will be no transmission to further generations. That happens in an individual after the zygote is formed. Only mutations occurring in a gamete or their precursors can be transmitted to the offspring. So if a mutation occurs at the gamete level, then it will be transferable. In addition to the gross structural chromosomal changes, submicroscopic alterations, including substitutions, that is replacement of single nucleotide by another, deletion, loss of one or more nucleotides, and insertion, addition of one or more nucleotide is basis of the mutation. Single base substitution is referred as point mutation, and majority are either missense or nonsense mutations. So such mutations does not manifest in an individual and are called as missense or nonsense mutations. Two-hit progression to tumor formation. It is proposed that germline mutation of one allele of tumor suppressor gene was not in itself sufficient to lead to tumor formation. So there is a germline mutation in a tumor suppressor gene, but that single germline mutation is not enough to cause a particular tumor. Hence, in a target tissue, acquired mutations that are known as somatic mutations need to occur and lead to complete loss of function of that gene. So for a particular tumor requires mutation in the germline as well as a somatic mutation, and the two different mutations are required for that particular tumor, and this concept is called as two-hit progression to tumor formation. The concept has recently emerged from a number of disorders like neurofibromatosis 1 and tuberous sclerosis. Mosaicism. Mosaicism describes an individual with two or more cell lineage of different genotypes that are derived from the same zygote. Two different genotypes derived from the same zygote is called as the mosaicism. Then lionization. Lionization is a concept in which one of the two X chromosomes in the cell of normal females undergo inactivation at the early stage of embryonic development, that is 12 to 16 days after fertilization. So this phenomena by which one of the X chromosome become inactive is called as lionization. The inactive X remains condensed as a densely stained mass of chromatin, which is known as the bar body. For each somatic cells, it is random whether the paternal X or the maternal X is inactivated, but the choice is fixed for all the subsequent descendants of that particular cell. So in such cases, an individual is a mosaic that would be carrying two different types of X chromosomes, one with different characters, two different types of X chromosomes, in which one is lionized and other is, in other pattern, the other X chromosome is lionized. Blaschko lines. As a result of lionization, the heterozygous state of various X-linked gene defects may give rise to mosaic pattern of cutaneous lesions, which confirms to the system of lines on the body that are described first by Blaschko in 1901. The original description of Blaschko refers to a system of lines on human skin which the linear nevi and dermatosis follow. Many genetic skin diseases reflect mosaicism with mutations occurring after fertilization, post-zygotic. In general, the greater propensity of cells that are abnormal, the greater is severity of resulting phenotype. So the mutation is a somatic mutation and is occurring post-zygotic stage, depending upon when the mutation occur and number of the cells that are derived from that particular mutation, the severity of the disorder manifests. So all the cells will not be defective. Hence, the disorder will be called as mosaic and it will manifest in various degree of severity. It is likely that all nevi reflect the genetic mosaicism. Streaky or patchy pigmentary skin changes may be a clue to the presence of mosaicism. These are the Blaschko lines. Gonadal mosaicism and recurrence rate. An autosomal dominant trait may occur in more than one sibling, even though both parents are apparently unaffected. As we described the rules of autosomal dominant inheritance, we said that in all the autosomal dominant diseases, one of the parents should be suffering from the disease. But this may not happen. And this phenomena may be explained by the gonadal mosaicism due to presence of mutation in germline, but not somatic cells. So the mutation occurred in germline. Both parents are normal, but at the level of germline, a mutation occurs, and that mutation is then transferred to the next generation, and the parents are absolutely normal. Evidence of genetic mosaicism exists in tuberous sclerosis, in bullous ichthyosiform erythroderma, and neurofibromatosis 1. So if you find a particular person suffering from tuberous sclerosis, neurofibromatosis 1, or bullous ecdysiform erythroderma, in which none of the parent is affected, then you should be able to detect that it is because of mutation in germline, as a result of which individual is affected, but parents are normal. Genomic imprinting, the phenomena whereby the DNA sequence derived from one parent acts in a different way compared with that derived from the other parent. The molecular basis of imprinting is thought to be methylation, the process whereby a methyl group, which is CH3, is added methyl group CH3 is added to DNA nucleotides, typically cytosine. Methylation is usually associated with reduced level of expression of a gene. So the phenomena is that a particular DNA sequence manifests as a different, manifests in different ways in one parent as well as from the other parent. And it is because of methylation of cysteine amino acid, as a result of which that particular gene in which the methylation has occurred will not manifest and will not play a part in DNA sequencing, which results in variability in genomic expression. Uni Parental Diasome. Rarely, uni parental diasomy may occur when an offspring receives both copies of chromosome pair from one of its parents. So the chromosome, the gene from the other parent is not received, and both the genes are received from one parent. For some chromosomes, this results in recognizable phenotypes. For example, uniparental diasomy of maternal copies of chromosome 15 leads to Prader-Willi syndrome. Twin spotting, also called as diadomyosis, didymosis. Twin spotting or didymosis. The genetic concept of somatic recombination due to post-zygotic crossing over results in two homozygous daughter cells representing stem cells of two distinct types of nevi. So the problem is post-zygotic and of somatic recombination, and resulting in two homozygous cell patterns that results in two different types of nevi. For example, coexistence of telangiectatic nevus and nevus animicus in one person, and particular diseases in which multiple different types of nevi are seen that include phacomatoses, pigmentovascularis, and Proteus syndrome. Genetic linkage or linkage disequilibrium. Alleles at a gene locus residing close to each other on same chromosome remain linked in transmission so long as the chromosome remain intact. However, during reduction division or meiosis, such linkages may be disrupted if crossing over occurs. The closer two gene loci are situated on a chromosome, the less likely that they are to be separated by crossover and more likely that they are to be inherited together. So if I explain this phenomena, there are, for example, there are two genes for two different phenotypic characters, and the two genes are located close to each other. Then we expect that the closer the two genes are located with each other, there are less chances that these two genes will be separated from each other when the reduction, division, or meiosis occur during crossover. If they are spaced more, then there are chances that these two genes will be separated in further generations. When two alleles occur together more frequently or less frequently in a population than would have been expected from individual allele frequencies, they are said to be in a linkage disequilibrium. The linkage disequilibrium is one of the important causes of disease association. For example, Nail-Patella syndrome is a clinical example of a disorder that shows a genetic linkage to blood group ABO locus on chromosome 9. So the two loci are so closely related that in every generation, these two will be linked together, and this is a linkage disequilibrium. Human histocompatibility antigen and disease association. Human leukocyte antigens are glycoproteins on cell surface of most of the nucleated human cells. These differ in subtle way from person to person, just like the fingerprints differ from each person. These fingerprints allow person's immune system to recognize a given cell as its own. The HLA region is located on short arm of chromosome 6 that is referred to as a major histocompatibility complex. A person inherits HLA as a set, one set haplotype from each parent. There are at least 4 or 5 gene loci that produce HLA, which are termed as A, B, C, D, and DR. The association of HLA with a given disease means that there is a higher incidence of HLA antigen in a group of patients with the disease than in a group of people without the disease. So this is called as the HLA association. If a particular disease, individuals carry that HLA antigen more frequently than the other one. How HLA is involved in pathogenesis of a disease. There are different phenomena, the first of which is the molecular mimicry. An infective agent may have similar configuration of the HLA so that the agent is not attacked by the body defenses. Alternatively, the agent might differ only slightly from the HLA so that antibodies are produced against that particular antigen, which is also attacking the cells containing that similar HLA pattern. So this is the basis of autoimmune disorders. Genetic linkage. HLA may be close to another gene on the same chromosome that produce a disease. This is example of genetic disequilibrium. Association between HLA and particular disease is rarely absolute. So ankylosing spondylitis is one such example where there is a strong association with HLA-B27 antigen. But some patients with this disease are B27 negative, and there are many patients with B27 positive, but does not manifest ankylosing spondylitis. Receptor effects. Many chemicals, including drugs and toxins, bind to cell surface before they are taken up into the cytoplasm. Since HLA are present on the cell surface, they could modify the binding of these potentially toxic substances, and specificity for such binding may be determined, at least in part, by DNA variation of HLA molecules. So there will be variable effect of potentially toxic substances determined by particular HLA molecules. Now, in the end, I'm going to discuss the few salient features of genetic counseling. This is a step you will be carrying out in your OPDs and even in exams for all the genetically transferable diseases. What is a genetic counseling? It's a counseling strategy and it's an educational process which assists an affected person or person at risk in understanding the nature of the disease which is in question, its mode of transmission, whether autosomal dominant, autosomal recessive, X-linked recessive, options available for treatment, and possibility of prenatal diagnosis. Situations encountered for genetic counseling. That is a prenatal diagnosis, when you have to make a prenatal diagnosis for a particular disease that is running in the family, or the diagnosis is made at the time of birth. Now you have to counsel the parents about that particular child and the future children. A disease with adult onset, sometimes it is required, and coupled with the family history of a disease. What are the steps in genetic counseling? First of all, explain the nature of the disease in detail, then the natural course of the disease, its best and worst prognosis, the transmission of that particular disease, whether it's autosomal recessive, autosomal dominant, X-linked recessive, or X-linked dominant. Then all the available treatment options, medicines, and the support groups for that particular disease, chances of prenatal diagnosis, screening, and options for abortion in subsequent pregnancies, and continuous counseling with recent advances. So I thank you all for patient listening. This was indeed a difficult subject, and I welcome all the questions which you can ask me directly on my WhatsApp number or even in the WhatsApp forums. Thank you and have a good day and a nice weekend.